So my disclosures are that I work with a drug company, Novartis, that has licensed this technology. I have research funding through them, and I consult for them. So on behalf of the folks at Children's Hospital in Philadelphia and also the group at the University of Pennsylvania, I appreciate the chance to sort of talk about our data. I will talk about acknowledgments at the end of the talk, but I want to start by acknowledging the person at Penn who's really driven all of this, and that is Carl June. And so what I want to do today is talk a little bit about where we are, talk a little bit about the preclinical data that we use to drive development of chimeric antigen receptor T cells, some of which occurred in my lab and a lot of which occurred other places, and then spend the bulk of the time on clinical data and give you a little bit of a sense of the various approaches that are being used to implement this technology. But I think the main thing that I want to convey in this talk is that in the last couple years, the whole immunotherapy field has completely changed. It's not just CAR T cells, it's not just monoclonal antibodies, BiTEs, it's also checkpoint inhibitors. I mean, this is completely changing the landscape. And so let's talk about landscape. I was sort of inspired by Archie's slides last night to talk a little bit about big picture stuff. And so let me take a contrarian view to what everybody always says about pediatric oncology. And so everybody shows this slide, right? And so I don't even need to describe it to this group, but it's really, this is the triumph of sort of iterative, cooperative, large, phase 3, especially clinical trials that sort of figured out how to improve outcome in kids with ALL. And the result of this process, at least in pediatrics, and the data about AYA, I think is extremely important, but at least in pediatrics is this terrific outcome in patients. But as you know, there are patients that we don't serve with this approach. And this is really making chemotherapy work better. This is old drugs. Peter Adamson has this awesome slide where he shows the FDA approval dates of all the drugs that we use for ALL therapy, and they're all, many of them are older than me, and all of them are older than our residents, and it ain't a new drug triumph that gets you from here to here. This jump up in terms of CNS control is something that I'll return to very briefly. But there's another way to look at the data, and this is where we are now. So this is U.S. cancer mortality, and what you see is this, the impact of this approach is in this line here, where you see this linear decrease in cancer mortality from the mid-70s to the late 90s, again in pediatrics, again maybe leaving some of the AYA patients behind. But what you see all of a sudden is that somewhere in 1998, this stops working, okay? So if you take this line and you extend it out and you ask when do we hit the x-axis and we're done and we have completed our task of controlling pediatric cancer mortality, it's 2006. But that ain't what happened. So where we are now is this part of the curve. So iterative changes in chemotherapy, improvements there's the ability to allocate patients to therapies based on their risk, the ability to control toxicities, which has been such an important part of the discussions we've had, all incredibly important. But how are we going to get down to zero at this rate? Well, again, Peter Adamson did this calculation for me. It's going to take 150 years. So that's where we are now. We're on this part of the curve. So chemo ain't going to cut it if we want to get back to any improvement in long-term disease-free control in patients that we aren't serving. And the patients who are left behind by the awesome control of disease that we've achieved in ALL are a mess. So as we've gotten better at treating most of the patients, the smaller and smaller group of patients who aren't served by this strategy get worse and worse. And we see this, I'm a transplanter by trade, and so we see this in transplant. So the patients that we're trying to transplant are in worse shape and have worse disease and are harder to control and harder to get into MRD positive than patients were previously. And there was an interesting discussion yesterday about the impact of MRD on the decision to go to transplant. And do we know that beating somebody into MRD negativity with multiple cycles of chemotherapy is the same thing as getting there quickly? We don't know that. We have no idea. So we really are in a different era. And so this is just data that shows the miserableness of the outcome in one group of patients for second and greater relapse. We can get these patients into remission part of the time, but we really can't control their disease. So all of this it's probably worth exploring what we're going to be talking about for the next 2 hours. So my subject today is cellular immunotherapy of cancer. And so what I want you to think about are the things that we're good at and the things that we haven't been good at. So for about more than 10 years, we've sort of figured out how it is that we can take T cells that can't see cancer cells and force the T cells to see cancer cells. And so we've used approaches of engineering T cells. We'll be talking about chimeric antigen receptor T cells. There are also engineered T cell receptors. They have their pluses and minuses for both of these approaches. I don't need to get into that today. Happy to answer questions if they come up. But targeting, we know how to do. Now, the other thing we need to do is we need to make these cells. We have to make cells for each patient, and that's complicated. We need a GMP, a good manufacturing practice compliant approach. We need to make these cells expand ex vivo. We can do all those things, and we've been able to do them for years. The problem we've had is what happens when we put these cells in patients. And so these are the things in yellow where they haven't worked until very recently. So we want to see these T cells expand. We don't want to see them disappear. We know how an effector T cell response works. The way it works is that the cells have to proliferate like mad. You go from a very small number of precursor cells into a very large number of effector cells, and then you have a memory response. So that requires enormous doublings of T cells. And so you want to be able to do that if you want to retain the power of the T cell. So we need T cells with a younger phenotype. We need T cells that aren't exhausted for this expansion. So we'll talk about expansion a lot. And then we'll talk about persistence. So if you want a long-term effect, if you want to control disease over years and not just weeks, you need persistence, and this probably requires some degree of memory. And the group at Seattle has done some amazing work, and that's been followed up by a lot of people on the issue of using central memory type T cells for this. We need an adequate effector to target ratio. That requires expansion. And at this point, my argument is that we need evidence of efficacy in phase 1. So we know what efficacy looks like. I'll try to show you what it looks like today. So that's what we ought to be looking for in thinking about these expensive strategies, these complicated strategies. We need to be looking for efficacy and not just safety in our phase 1 trials. That's a personal opinion. So these chimeric antigen receptors come in generations. This is probably too immunotechnical, but I'll very briefly comment on this. So first generation is what was used for quite a while in a number of clinical trials, which just involved an activation domain for T cell activation, the CD3 zeta domain. That activates T cells, but we know something about T cells that require a second signal. That's a costimulatory signal. So I think an important innovation was the addition of a costimulatory domain, a single costimulatory domain, and that created the second generation CARs, which are really the CARs in clinical trials right now that are producing the dramatic clinical responses. You can extend that concept to third generation by putting more than one costimulatory domain. We don't know if that's going to work better, but it's certainly a concept that exists out there. There's no clinical data to tell us how that's going to work. So this is our approach with the chimeric antigen receptor. So what you have is a tumor cell or a normal B cell, we can't tell the difference, that expresses CD19, which is a great target in B cell malignancies other than myeloma. And so this is the tumor cell, this is the T cell. The T cell has this artificial molecule. The outside of the artificial molecule is basically a single chain FV, a piece of an antibody. So in theory, anything you have a monoclonal antibody to, you could build a CAR from, as long as you are okay with the cell bearing that antigen being destroyed, which is a major safety issue. Why does everybody do CD19 CARs? Because CD19 is just on B cells, and you can live without your B cells. On the inside, you have this activation domain, and then you have these costimulatory domains. A lot of groups have focused on CD28, which is a very well-known costimulatory molecule. We focused on a different costimulatory molecule, 4-1BB. Again, we can talk about that. It's not something I want to spend a lot of time on. So this is how this process works, and I'm sure you're all familiar with this. But basically what happens is that we take T cells from the patient. They do have to have some T cells, by the way. We need patients who have an absolute lymphocyte count of around 500 to be able to do this. We take T cells from the patient, and we basically expose those T cells to a virus. The particular virus that we're using, a lot of groups have used retroviral vectors. We've used a lentiviral vector. I'll get back to that issue at the very end of the talk. We permanently modify the genome of that T cell, so this is inserted randomly in the genome to put in this CAR gene, this CAR transgene, which then is expressed on the cell surface of the T cell. The endogenous T cell receptors remain in place, although we think perhaps that the very strong signaling from the CARs overrides any signaling from the endogenous TCR, but we don't actually know that. This drives an interaction with a cancer cell, which hopefully kills it, and I'll show you what that looks like. So that's the manufacturing process. It takes about 3 weeks to do this, 2 weeks of culture and 1 week of release testing. I think an important part of this is how these cells are actually made. I said ex vivo expansion is an important part of this. We've been able to do this for a long time, but we do it a little differently. We use these beads. So these beads that have antibodies that bind both CD3 and CD28, which provide both signal 1 and signal 2 to these guys, which are T cells. And so this provides a very strong stimulus, but it maintains the young phenotype of the T cell. So we think that the T cells that come out of this manufacturing process are enormously expanded, but can continue to grow in the patient. And I'll show you some clinical data that shows you that I'm not just making that thought up. I think there's less replicative senescence. I think there's less exhaustion. I think there's more induction of telomerase. Those are all things that go along with T cells that have more of a capacity to reproduce. So this is what this looks like. So this is a video of a human cancer cell being attacked by an engineered human T cell. And the gray cell at the bottom here is our T cell. These green cells are our cancer cells. And so what I want you to look at when I start the video is what happens to the cell membrane as this guy docks with this guy. And so what you're seeing here is the interaction. T cell comes up, interacts, and then this bubbling here, I don't know why it keeps stopping, it's a mystery to me, is the destruction of basically destruction of the membrane, the T cells poking holes in the cell membrane. So this is a highly resistant cancer obtained, it's an ALL obtained from a pediatric patient, very chemo-resistant, but the resistance patterns of having holes poked in your membrane are different than chemotherapy resistance. So there's a non-cross reactivity for this approach. Little bit of preclinical data. I beg your indulgence for this, but people often ask why we chose a particular approach that we did with the costimulatory domain that we did. And basically, this is just a mouse model of a xenogeneic mouse model where we have human T cells, human ALL, primary human ALL engrafted in the mouse. And we asked a simple question, how did the T cells grow? If you just put in T cells, if you use these various costimulatory domains, and this is the one we're using clinically, and you can see there's a really big difference in terms of proliferation, at least in our hands. And other folks who believe in other costimulatory domains will show you data that look like this, except it comes to a different conclusion. I don't know why. Don't see a real big difference in persistence in any adequately activated T cell in this mouse model. So I want to briefly comment on this issue of central memory. So for folks who are not familiar with the way we divvy up T cells these days, they're naive T cells that haven't seen antigen. They're terminal effector cells. These are the bumblebee cells that are actually killing a few cells before they go away. The effector memory cells, and then this very important cell, the central memory cell, which really has the phenotype to stick around for a long time. And so in our mouse models, we were curing mice with human leukemia using engineered human T cells, and we were asking ourselves about these T cell phenotypes. And so you see when we collect T cells from a normal human donor, you have some effector memory cells, you have a lot of naive cells, and you have a very small number of central memory cells. When things are really moving and the T cells are killing the ALL actively, it's almost all these effector memory cells. Sorry, I skipped one step. So we have these effector memory cells after the culture. Naive T cells have disappeared because, of course, all the cells are activated, so they're the effector memory phenotype. Still have preservation of some central memory cells. And then when things are really moving in the mouse and we're killing the disease, you have these terminal effector cells, a small number of effector memory cells, and still a small number of central memory cells. When everything's calmed down, the mouse is cured, or at least cured out to a year, which is how long we follow these mice. Then you see that most of the cells that remain are these central memory cells. So this formation of immunologic memory may be part of the phenomenon that we're seeing. So let's switch. So Lenny mentioned that he ran into one of my patients. I talk about this patient a lot. She's the first patient that we treated, and this young lady. So we learned a lot from this young lady, and so we talk about her a lot. So she was she's a typical kid that we have to deal with. She's the flip side of all the success. So she has standard risk ALL. She's MRD negative. God forbid somebody who initially treated her told her if you had to have cancer, this is the kind of cancer to have, which I really tried to talk my fellows out of saying. Unfortunately, as these things go, we wouldn't have predicted that she was going to have a problem, but she did, as so many of the patients who do have a problem fit into this category. So she relapsed. So now she comes from her home center to us for a bone marrow transplant. Now, in pediatrics, we don't transplant patients with leukemia with active disease. So we put her back into remission. We're looking for a donor because she doesn't have any brothers or sisters, and it takes more than the month. We're working on it, and she relapses again. This happens. And so now she's resistant. So she gets a couple cycles of chemotherapy, including a clofarabine-based regimen. She's enrolled on this clinical trial as our first patient, first pediatric patient, first ALL patient. Imagine describing that to the parents. And when she comes in for her T-cell infusion, she's pancytopenic but has 60% blasts in her marrow. So clearly chemotherapy refractory. She does not get lymphodepleting chemotherapy because she's already plenty lymphodepleted from the clofarabine VP16 Cytoxan she got 6 weeks prior. So we give her T-cells. We give her 100 million T-cells per kilo. 11% of them are CAR positive. The rest are not. And I'll get back to that number because it's important. She got very sick. And I want to talk about why that was an issue and what we did about it. And then she entered a complete remission, which she has maintained a complete remission with no further therapy for 2.5 years. So what did we see? And we're seeing this over and over again. Well, first, I said proliferation matters. So here's what proliferation looks like. So this is flow cytometry. Most of the prior clinical studies picked up cells by PCR because they couldn't see them by flow. PCR is much more sensitive than flow, as of course we've heard from the MRD folks. So these are T cells, CD3 positive cells. These are cells that express the CAR. This is a day after we gave her T cells. 100 million cells, they all disappeared. Where did they go? Well, either they're gone or they're in the tissues. Turns out they're in the tissues, and as they proliferate, they pop back out into the peripheral blood. And now we're seeing that 2/3 to 3/4 of the T cells in this patient are these modified T cells. Now, that's interesting because we put in 11%, we got out 70%. What does that mean? That's antigen-driven T cell activation. It's not homeostatic expansion in a lymphodepleted patient. That would expand the whole population. This is antigen-driven. This is not just, we're showing this because it's the best one. We've treated 40 patients, and most of them are like this. In many of these patients, half their circulating white cells are gene-modified T cells. So it really is a difference in terms of proliferation. Now, I'm going to get into the issue of persistence in a lot more detail, but we have persistence now out not just to 18 months. I keep meaning to update this slide. I apologize, but out to 30 months. And in patients who undergo a complete response, the persistence is much longer, and I'll show you what this really looks like, than patients who don't have a complete response. So persistence sort of correlates with activity. That's not too terribly surprising. But we're seeing longer and long persistence in these patients. The first patient is out to 2.5 years, still has T cells. The shortest we've seen is one patient who I think probably had a rejection event and lost their T cells after great proliferation in just a couple weeks. That's an outlier, and this is the furthest we've gone. So the actual infusion of these T cells is no big deal. So this actually happened yesterday. So somebody texted me this picture, and I wanted to share it with you with permission from the family. So this young lady just got her T cells and obviously is not having a problem in the moment. But people can get sick, and I'll talk about what that is. So the infusional part can happen in the outpatient clinic. It's just you walk in with a syringe, 30 cc's of cells, inject it with a little bit of pre-meds, and it's over. That part is easy. It's really what happens after that matters, and I'll get into that in very significant detail. So one of the things that we've run into is this issue, and you'll hear about this again from the Blinatumomab folks, is the issue of cytokine release syndrome. So this can result in substantial toxicity, and did in our first patient. So when we looked at cytokines in these patients, this is a complicated slide, but basically what I'm showing you is a patient in blue who didn't respond, a patient in red who actually I saw last week who had a complete response with a mild cytokine release syndrome, and then a patient who had a complete response with a terrible cytokine release syndrome. And what we see in the green curve, which is the bad one, well, we see interferon gamma go up. We see interleukin-2 receptor go up. In some patients, we see GM-CSF go up. Not all that surprising. The thing that was surprising is IL-6, and that's this curve. And this is a log curve here. So this patient has a peak IL-6 level of 50,000 picograms per mil. That's not typically something you see in human beings. So very, very high IL-6. So that was really astonishing to us, that it wasn't expected. Didn't come out of the mouse models. Turns out not to be produced by T cells. It's just something that shows up when you put it in people. So if you look at this, and I'll show you another analysis of this, patients who have a complete response have pretty high IL-6 levels. Patients who have bad CRS have really high IL-6 levels, and patients who have a complete response and a very mild cytokine release syndrome, they have very low IL-6 levels. So there's definitely, and I'll show you the update on a lot of patients with this. So IL-6 matters. Turns out, and folks may be familiar with this story, that there's a drug that targets IL-6. I'd never heard of it. I'm not sure I could have spelled this drug 2.5 years ago, Tocilizumab. It's used by our rheumatoid arthritis colleagues. It has a pediatric label indication. It has a pediatric dose, unusually. So we actually had it in our pharmacy when we realized that IL-6 was so high in our very sick patient. It's typically given monthly, but we just give it once, or if sometimes a second dose a day or two later. Side effects are mild, and we use the label dose. So this IL-6 blockade medicine has really revolutionized the management of toxicity, and I'll tell you about that. So these are counts in our young lady. So she comes in pancytopenic. This is her white count, and these are her lymphocytes. We give her T cells. She's admitted to the hospital with a fever. She got them in the outpatient clinic. By Sunday, after we gave her the T cells on Wednesday, she's already up in the ICU with a temperature of 41 and hypotension. And she continues to get sicker. We give her steroids as per protocol. Doesn't make any difference. She's as sick as you can possibly be. 3 pressors, nitric, 100% O2 on an oscillator. So we thought she would not survive this. Gave her a Tanacept. Why? Because there was COG data that I was aware of that might control cytokine-induced lung injury. Didn't seem to do anything, although we didn't give it much time. On a Wednesday afternoon, we hear that her IL-6 is super high from a research lab, and yes, I know I'm not supposed to use research data to drive patient care, and we do not do this, but in this case, it was life-saving for this patient. They called me up and they said, IL-6 is through the roof. And so we give her this drug, Tocilizumab. In hours, she's better. She's off her pressors within 24 hours. All night, they spend with her hypertensive because they can't wean her fast enough. She goes from a white chest x-ray to a normal chest x-ray. She goes from 100% O2 to room air. She's on the vent for a few more days because, of course, she's paralyzed and sedated for the oscillator, and then she needs to get off of that. Full recovery, and remarkably fast. So the guy who runs our ICU at CHOP, Bob Berg, says this is the fastest he's ever seen a patient that sick get better. So this is fascinating to me. As you guys probably know, we tried cytokine, we didn't, the ICU folks tried cytokine blockade in sepsis, and it didn't work. And the reason why is probably this is a network effect, and you have to interrupt the network at multiple nodes to get an effect. But this appears to be a classic feedback loop, and IL-6 is necessary for the feedback loop. And if you interrupt IL-6, you interrupt the feedback loop, and the toxicity falls apart. And so we've seen that over and over again. So this was very, very interesting to us, that we could control the toxicity response using IL-6 blockade. So folks are going to be talking about blinatumomab. I'll talk about one blinatumomab patient with apologies and also contributions from my Amgen colleagues. So this was a young man who was treated on the pediatric phase 1 above the MTD. He got 30 micrograms of blinatumomab. He was given blinatumomab, looked just like our first patient up in the ICU, very sick, cytokine release syndrome, hypotensive, on an oscillator. Stopped the blina, of course. Didn't really make him much better. Recognized that his IL-6 was quite high, although not the crazy levels that we see in some of our patients. Had a prospective plan to intervene if this happened with tocilizumab, gave him tocilizumab, and he got better very quickly. So this cytokine release syndrome is not just limited to our particular brand of CAR cells. This happens over and over again, and IL-6 may be important across the space. So colleagues at NCI have used this approach. Colleagues at Sloan have used this approach. So clearly this is something that I think folks should probably be aware of. So that's great. Got that patient in remission, controlled their toxicity. But there are issues that we have to deal with. So this is the second patient, actually, that we treated. And we've seen a couple patients do this. So this young lady comes to us. Now, she's had a transplant, a cord blood transplant. She has relapsed. She's gotten blinatumomab. Didn't do anything for her. She comes to us. She has this MRD. So we talked about doing MRD by flow. That's what we do. And we'd love to do high-throughput sequencing MRD, but right now we're doing it by flow. Brent Wood does this beautifully for us at the University of Washington. So here are all the cells in the marrow, and here are various ways of looking at B cells. And I'm not sure that you need a PhD in flow cytometry to assess the difference between before we gave the T cells and after we gave the T cells. So here are bone marrow cells, but there are no B cells in this patient's marrow, neither malignant or normal. And of course, our colleagues at the MRD lab are like, what the heck is this? And we've really worked with them to try to understand how you analyze these specimens when you have no CD19, which is what you typically gate on when you do these analyses. So complete elimination, scorched earth of the B cell compartment after 1 month. And this is what we see in all of our responding patients. So that was the good news, but the good news didn't last. So this is research flow on this young lady's tumor, and we see her CD19 positive disease, and we see this little shoulder here. I mean, what's that? Is it negative? Is it dim? Is it really leukemia? Did we gate right? We don't know. Unfortunately, these cells all go away. These cells continue to grow, and she relapsed between 2 and 3 months with CD19 negative disease. So we've had a couple of these patients who've experienced CD19 negative relapse. So whether the blinatumomab beforehand added to the risk of this, I have absolutely no idea. When we've seen this, sometimes the patients have gotten prior blinatumomab, but in one case they did not. So we see this issue of CD19 recurrence, and although it's not in a large number of our patients, it's unquestionably a real phenomenon. Interesting point. So we see these cells in the blood, we see these cells in the marrow. We don't know if we see them in the tissues, but we clear tissue disease, so we think we see them there. But it's probably important whether they get into the CSF or not. So these are large granular lymphocytes that actually are CAR cells. These are not blasts. And the heme technologist who reviews all of our slides can actually see these cells before the flow guys do, and calls me up and says, I see them in the periphery, this patient is getting started, typically around day 7 after infusion, we see these cells in the blood. But what was surprising to us when we did routine taps, as we do in these patients, is that we see the cells in the CSF. Now, we don't know what they're doing there, okay? I'm not saying they're controlling disease. Right now, we're not treating patients with CNS3 disease because we're concerned about neurotoxicity. We're just opening a cohort to test this like next week, so maybe we'll have more data. We've had a couple patients with CNS2A disease that subsequently entered CNS remission. I don't know if the T cells did it or not. That's all the data that I have. But in 17 out of 19 samples where we were able to analyze this, we see these CAR cells. So they get into the CSF most of the time. And almost all the cells in these patients, their CSF white counts are all that high. 10, 15 maybe, but they're almost all CAR cells. So I mentioned QPCR as a technique for detecting these, Sue's heard this before, so she's heading out the back. So I mentioned QPCR as a technique for detecting cells, and this is just data I got off the web from a group that was looking at the performance of their cells after an infusion. And if this is hard to read, I can just point out what I need to, which is that the data are kind of noisy, and the peaks are around 2.5 copies of the transgene per microgram of genomic DNA, which is the way we look at this. So just remember that number, 2.5. So this is what it looks like when you can't get enough proliferation to actually pick up the cells by flow. And so if you want to use that technique, it's actually a great technique, of course, because it's so wonderfully quantitative. So this is what our data looks like. And this slide is busy, but I think we can make the point. So this is copies per microgram DNA. Every single patient that we've treated in this cohort of 30 that we're currently reporting. And the curves in black are responses, and the 3 curves in red are non-responses. And there's 2.5 down here. This is a log scale, 100, 1,000, 100,000, 4 orders of magnitude more proliferation. And I think that matters. I think that the groups who are seeing dramatic responses, not just our group, are seeing dramatic proliferation, very significant proliferation. And then this issue of persistence comes into play. So you can follow these cells over time. This is about the cutoff for flow, is around just below 1,000 copies. And below that, you really can't pick up the cells by flow anymore. But we can pick them up by PCR. And so we see there's a substantial number of patients who've maintained their cells for a long period of time, out to a year. And this includes the young lady that Lenny mentioned, who we treated first, who still has her cells out at 2.5 years. Goes down at every determination, but they haven't gone yet. And then there are a group of patients who lose their cells more quickly. And these patients are at increased risk of relapse. There's no doubt about that. So where are we with this? So these are the data that we're currently presenting. Over 110 patients at CHOP and Penn have gotten this. They started with CLL, where they saw dramatic responses, Dave Porter's group. We switched to ALL, including that first young lady that we've just talked about. They've started to do NHL, and the group at NCI has done a lot of patients with diffuse large B-cell lymphoma and see a reasonably high response rate. So we're able to see responses in all of these CD19 positive malignancies. Treated 25 kids. Our colleagues at Penn then started treating adults with ALL. And so out of that 30, 25 kids and 5 adults, the CR rate is 90%. And so 90% of these patients with relapse refractory disease, many after a transplant, and I'll get to that point, have gone into remission. Of this group, we've had 6 relapses, half CD19 positive and half CD19 negative. Follow-up is getting a little bit longer, but it's still short. Median is now up to 8 months, and the range out to 2.5 years. So that's where we are right now. So it's an important question to ask what the disease burden of the patients are at the time they were treated. And so this graph just shows that. This is gross disease. This is less than 5% blasts. And this is the 3 patients who actually became MRD negative after the lymphodepleting chemotherapy. And what you see is that most of the patients are in this bin. These red bars are the relapses. So, no way I can statistically say whether the relapse rate differs in these 3 groups at all. But that's where we are. We have 1 relapse here and a few more relapses here. So most of these patients were refractory to multiple prior therapies. Now, as a transplanter, this next slide is kind of interesting to me. So most of these patients have already had their transplant. So they came to us after stem cell transplant, after a relapse, and that's 2/3 of our patients post-allo, and then 1/3 of our patients who have not had an allogeneic transplant. That's what our numbers are running at right now. Now, this is an important point if you care about transplant. We collect the cells from the recipient. We don't collect the cells from the donor. So we did that on purpose. 2 reasons. One is I thought that perhaps these cells might be better tolerized in the recipient than they are in the donor, and therefore I'd have less risk of GVHD. And we've seen no GVHD in any of these infusions, so that's great. And we also don't treat patients with GVHD because we worry about waking that up with the activated T cells. And then the other point, of course, is that in the United States, the question of haplotransplant is very apropos, but in the United States, the of the unrelated donor population, half of those transplants are with cord blood. I ain't going back to those donors. So I really have to have an approach that's going to work across the board, and so we go to the recipient. So we have no experience with collecting donor T cells. Other folks have tried this, but we have not tried this. So they need to be 6 months out. They need to not have GVHD. We don't cause GVHD. And even though they've relapsed, there's the cells that we collect. The median donor chimerism in the cells that we collect is 100%. The cells that we get, the T cells that we get are still mostly donor T cells. It's not universally true, but it's true in almost all the patients. So I think that that's very interesting. No GVHD, and we can collect these donor-tolerized T cells from the recipient. So in addition to the cytokine release syndrome, we've also seen this issue of macrophage activation syndrome. Again, I didn't pick this up in the mice, was not predicting this. So we were checking ferritins in these patients, and we saw ferritins that were crazy, 16,000 to 400,000. That particular number, 400,000, broke the analyzer. The Siemens people had to fly out to our clinical lab. The clinical lab hates these patients because they have to dilute and dilute and dilute until they can get a readable number to get these very high ferritins. And this is very suggestive of a macrophage activation syndrome, or what we would call hemophagocytic lymphohistiocytosis, or HLH. They have coagulopathies that are universally very high D-dimers, low fibrinogen in some patients, bleeding in very few, hepatosplenomegaly in some patients, transaminitis. Hematophagocytosis is there, but it's not rampaging. You can't pick it up on slides unless you do a CD163 stain. It goes away with cytokine blockade. Interestingly, a couple of the patients that we treated, this first kid and a subsequent kid, both had grade 4 cytokine release syndrome. They have predisposing hypomorphic perforin alleles that have been associated with secondary HLH. So maybe these perforin mutations are part of this story, but they're not the whole story, because we see a lot more cytokine release syndrome than just these two kids. So what are our markers for this? So I said ferritin was important. Remember, this is log scale here, all right? 1,000, 10,000, 100,000. And so we see patients who do and don't have, so don't have CRS, bad CRS, do have bad CRS. Bad CRS means you go to the unit and you get at least one pressor. And what we see is a very significant difference in ferritin. C-reactive protein has been proposed by the Sloan group. We see a very significant difference between patients who don't go to the unit and do. Although we do see some patients with very high CRPs who don't go to the unit, including these open circles, a couple non-responders. So I don't know how predictive this is. That's going to require a little bit more modeling and people who are way smarter about predictive biomarkers than I am, because this is all a retrospective analysis. But clearly there's an association between high CRP and severe cytokine release syndrome, this, as a marker of inflammation. Now this is a key point that I'd like people to actually come away from this talk with a thought about. So here's the disease burden, and it's just bone marrow blast percentage. Okay, you can do all sorts of great modeling in CLL patients in terms of what their disease burden is. You can do cross-sectional imaging, but just as this rough suggestion, the day before we give the T cells, we do a bone marrow, and we see their bone marrow blast percentage, and you see the difference between the patients with high blast percentage and with low blast percentage, and the cutoff is around 50%. So very few patients. Patients who are below 50% don't go to the unit. Patients who are above 50% have a high chance of going to the unit. So if you think about deploying this therapy earlier in patients with lower disease burdens, which ain't what we're doing right now, we're going to be here and not here. And we're not going to see the same dramatic toxicities that we see in a third of our patients. Cytokines, again, I've made this point. Here's interferon gamma as an effector cytokine, huge differences between disease burden and CRS. And this is really a disease burden, how hard the T cells have to work to get the disease under control. And then again, this 2 order of magnitude difference in interleukin-6. This is retrospective. We're not getting interleukin-6 in real time. Maybe you guys can get that out of your labs, but we send it to ARUP and it takes us 4 days, so that we're not using the numbers. We're making a decision about treating patients clinically and not using cytokines to actually decide what to do. But we see the effector cytokine, we see the toxicity cytokine, and it associates very strongly with patients with high disease burdens and a risk of going into the intensive care unit. So let's look at the persistence data. And so this is what the persistence data look like. This is the persistence by flow, which is less sensitive, but we see that more than 2/3 of these patients still have their T cells at 6 months, so that's a good sign in terms of persistence. What's interesting is I don't actually need to measure the T cells. I can measure the B cells. If the patient has no B cells, they've got T cells because the T cells are killing all their B cells. I hope that made sense. So B-cell aplasia is a very good pharmacodynamic marker for activity of these engineered T cells. And so these blue circles are no B cells. And so you see what these patients look like. And the red crosses are, we see a few B cells. This was an outlier. Patient remains in remission and has no B cells and remains dependent on immunoglobulin. But there are patients who lose their T cells early, get their B cells back, and both of these patients experienced a relapse. So if you lose your T cells before 3 months, the risk of relapse goes way up. It's not 100%, but it goes way up. But you see a lot of these patients who are maintaining their B-cell aplasia for a long period of time. So what are our data in terms of overall survival and event-free survival? Here we are. So this is the overall survival because some of these patients are salvaged with subsequent T-cell infusions or other therapies. So the 6-month overall survival is 78% in these patients. Most of these patients don't go to transplant, and that may be something I get pilloried for, but that's just the way it is. A, because they've already had their transplant, or B, because the parents elect not to go. We tell these parents, if this is their first CR in ALL after a refractory course, that it's standard of care to go to transplant. And then that's what we told the parents of the first kid that we treated. Now, this kid's going to school. She's got her hair back. She feels great. We still have T cells, we still have B-cell aplasia. We don't know what the performance characteristics are. And the family says, not so interested. I don't know what to do about that. But they don't go to transplant. So we only had a couple of the patients on this curve actually go to transplant. So toxicities, B-cell aplasia is a real toxicity. They need IVIG replacement. This is not Rituxan. They need IVIG replacement because all their B cells are gone. We really don't have a problem with tumor lysis syndrome. This is really seen in the CLL patients where they have much higher disease burdens. We see the cytokine release syndrome. 1/3 of our refractory patients go to the ICU. 3/4 of our patients with very high disease burden go to the ICU. They're there for anywhere from 3 to 10 days, but we can control this with the IL-6 blockade. And we can predict this in terms of the tumor burden of the patient. So for patients who have low disease, I think it's nothing like this. We see this macrophage activation syndrome. I didn't get into this, but we see neurotoxicity in some patients, which is entirely reversible, lasts 2 or 3 days, confusion and aphasia. Very, very striking, but it goes away without treatment. So where are we? What's the potential here? So I think we can control patients who have MRD. I think if a patient recurs, I think we have good data that we can get them back into remission. And we talk about this need to produce an MRD negative state prior to transplant. We can do that. So the concept here is this a bridge to transplant? And this is going to be an issue that's explored in the subsequent talks as well. We're initiating trials. We've just enrolled the first 6 patients in a multi-site trial in pediatric ALL. I'm personally delighted to say that pediatric ALL is the first indication that the drug company Novartis is seeking for registration. Well, that's awesome. That doesn't happen too often. And so this is really, because of the high response rates that we've seen, this is really leading the way for this particular drug, if you want to call it a drug, and the drug company does, in ALL. So the broader question, I'm going to get myself in trouble with this audience, so I'll apologize ahead of time, but is for patients who already had a transplant, the idea of going back to transplant is, we do it, I do it, but it's not that satisfying. The outcome after, this is adult data, but the outcome after a relapse post-allo, regardless of what you do, is very bad. So it's not that satisfying a modality. But also for patients who've not undergone transplant, a lot of them aren't going to transplant as we watch what the T cells do and whether they can remain in remission. I don't know if that's right or not. I've just shown you the data. So the real question in my mind is, can we replace transplant with these cells? We're not at that point yet. But for folks like you guys in the room, the decision is really, what data do you need to see that you'd be willing to consider that possibility? That's the only question I want to ask. When we think about this, what kind of persistence, what kind of long-term data, what kind of disease control? Half of patients relapse after transplant. We saw this in the 0431 study. What do you need to say, well, I might consider a study where I'm not sending my patients to transplant? Now, there are a lot of these studies ongoing. I've talked about our study, but there are some awesome data from Sloan. There's great data from the NCI. The group at Seattle is getting patients enrolled on trials as well. We don't know the performance characteristics as well. This is just a table that E.J. Spall and I pulled together of all the different, I don't want to go through this, believe me, I'm not going to do that to you, but all the different studies that were presented at the ASH meeting last year. There was a lot of excitement in this area, and we're seeing high response rates in a number of the different centers. Sloan published their data recently in Science Translational Medicine. This isn't just one place. We're seeing this issue at multiple places with multiple different technologies and with different degrees of persistence, I will say that. So just indicating that there are a lot of groups doing this sort of work, and a number of these groups are now over this activity threshold where we're seeing high proliferation and a hope for higher CR rates, and then a question of how long do you keep your T cells, which I think is really, really important. So bigger picture stuff. Lenny wanted me to put this in context. What are the issues that we think about? I won't go through all of these, but vector. So what virus do we use? Do we use a non-viral technology? The group at MD Anderson is developing Sleeping Beauty, which is a non-viral technology. Lenti, what does lentivirus mean? We call it a lenti, but it's a modified form of the HIV virus with all the infectious capabilities of the virus taken out. I mean, I got to explain that to patients when I talk about it. We want to know how we do the construct of the vector. Disease bulk, obviously that's an issue. We can control bulk disease. It doesn't just work in MRD. But we can control disease at high levels, but with some toxicity. What do we do before infusion? Do we transplant these patients after infusion? There are a variety of different issues. And then this issue of cell manufacturing, for insider baseball, we think that bead type cell manufacturing is very powerful. So we have this approach of these CD3, CD28 beads. The NIH has used, especially Rosenberg's group, has used OKT3 and IL-2. There are other ways to do this. The shorter the culture, the better. So it takes us 2 weeks to make these cells. It takes the NCI 10 days to make theirs. Can we do this faster? Release testing. Release testing is a week for me and faster for the NCI group because they don't use a lentivirus. So all of these issues, how fast can we get to treating these patients? What do we do prior to infusion, I think is important. Can we do this after stem cell transplant? You bet we can. Do we need a stem cell transplant? You tell me, I'm not sure. This issue of can we control disease in the CNS? Well, we're going to be enrolling some CNS3 patients, and I'll be able to report to you on this issue. Other sanctuary sites, we don't have any data about this at all. So I don't have anybody with active extramedullary disease. I've seen organ lesions, but I've not seen skin. I've not seen testes. So I don't really know about other sanctuary sites. This issue of B-cell aplasia, as long as you have these T cells, you will have B-cell aplasia. I think I've shown you that. And you need IVIG replacement. A lot of people ask about suicide genes, and that's an available technology. That's not something we're using right now, but I think it's a really good question what the role ought to be. If these kids really are going to be keeping their cells, their engineered T cells for several years, when do you say, well, I'd like to erase these T cells and get them back to not needing IVIG? They actually get Hizentra, which is a sub-Q preparation, so they get it at home, which is kind of cool. But I mean, they need it. And if we're going to do this for a lot of people, we have I'm not sure there's enough IVIG in the world. So we really do have to consider this issue. It's a real problem. So skip this. I don't think we need to get into this. So this is a slide out of sequence, and I've already described this. But basically, as a transplanter, my crazy idea is can we actually replace transplants? So this is the young lady that Lenny mentioned. Now 2.5 years out. This is a picture taken from her Facebook page with her parents' permission. My daughter tells me that 300,000 people saw this picture, and she was very jealous about that. This notion that we can get long-term responses in some patients without transplant is a very attractive notion to me as a transplanter. Because for 20 years, I've been doing family meetings, and I've always told the families, if I had a better way to do this, I would rather do it that way, because transplant is hard. We're not seeing GVHD. Our TRM, it's not like we're going to not see TRM, but we haven't, knock wood, seen it yet. So the TRM rate is probably relatively low. It's probably lower than transplant. Relapse rate, we'll have to see. That's, of course, the real question. So this has gotten a little bit of press. This is the kind of nonsense you deal with. If you Google HIV cures leukemia, you get 25 pages of this stuff. So no, it's not HIV. No, we don't give it to the patients. I can't convince anybody of that fact. But the reality is that this is a modified form of the HIV virus, and so there's a lot of interest in that aspect of it. This is an article that came out yesterday in the Wall Street Journal talking about CAR therapy, talking about Novartis and Juno, which is another company that's working with the Hutch and Sloan Kettering, Kite, which is working with the folks at the NCI. And what are the hurdles getting these into patients? The real hurdle is going to be cost, right? What's this stuff going to cost? I don't know. I have no idea. I'm not privy to that stuff. But if you look at what's coming out now, if you look at Solvati, the Hep C drug, it probably ain't going to be cheap. So that's an issue. So with that, and leaving a couple minutes, I hope, for questions, I just want to acknowledge all the people involved. I mentioned Carl June in the beginning, the real leader in cell therapy at Penn, the folks in my lab, Bruce Levine, who makes these cells, my UPenn colleagues, including Dave Porter, who's been such a great guy to work with, Noel Fry, who runs the ALL trials, the folks, the clinical staff at CHOP, the folks at Novartis, the people who do the research correlates. These are the people who got me the IL-6 data when I was screaming at them on the phone saying, no, I need it now. They got it to me now, and it really made a difference. And of course, what are you going to say except to thank the patients and the families who are willing. Brand new therapy, first time it's ever been deployed in kids. We're going to try this out. We really don't know what's going to happen, but I'll put my kid on a clinical trial. And you go back to the first slide I showed, 40 years of people doing that is really what we owe the ability to control ALL, is patients and families who are willing to go on clinical trials. And I think that's so important. And with that, I'll thank you for your attention and take any questions.